In this lecture, I will be discussing the processes of mitosis and meiosis that eukaryotic cells undergo to proliferate themselves. I have here a picture which I believe most of you would already be able to recognize. We can see here strongly staining bodies. This is viewed under the light microscope. And you might have guessed right. These strongly staining bodies, the dark colored ones, are the chromosomes. In fact, the word chromosome is derived from two words, chroma, which means color, and soma, meaning body. So they are the strongly colored bodies that we can observe under the microscope after we have dyed the cell. Then we can also see here quite faintly the pink colors, which are actually the spindle fibers, which are part of the cytoskeleton of the cell. So the spindle fibers are actually composed of microtubules, and they play an important part in cell division, particularly in mitosis and meiosis. Here we have an illustration of a eukaryotic cell. And it's very obvious already from this illustration how the internal parts of a eukaryotic cell are compartmentalized into membranous organelles. So these organelles actually have various functions of their own, but there are certain organelles that also function in cell division as well. So these organelles would include the nucleus, of course, which houses the genetic material which needs to be distributed during cell division into the daughter cells. And then here we can see the centriole. Okay, the centriole is usually just observed in animal cells, and they are usually housed also in a region, a dense region of the cell called the centrosome. Again, which is also primarily observed only in animal cells. And invisible here in the background are the cytoskeletons of the cell, composed of three types of molecules, or three types of structural fibers. We have the microtubules, we have the intermediate filaments, and then we have the actin filaments. But most importantly for mitosis and meiosis, the microtubules. But let's not forget, not all cells are eukaryotic. There are cells that do not have the nucleus or the membranous organelles that we have just observed a while ago. And these cells include the cells of bacteria and archaea, and they are known as prokaryotic cells. Now, because they lack the nucleus and the other complex organelles in the eukaryotic cells, they may undergo a process of cell division called binary fission, which is a lot simpler as compared to mitosis and meiosis. So we see here on the figure on the right that a bacterium cell contains a circular chromosome. The bacterial chromosome is circular, meaning to say there is no end. However, this chromosome, which is made of DNA, may be replicated faithfully by the bacterium itself. And once the two replicates have been completed, they may now be distributed into two new daughter cells through the process of binary fission, as we see here in the bottom portion. But eukaryotic cells have more than just one circular chromosome. In fact, eukaryotic cells don't have circular chromosomes. Okay? Their DNA are linear. The molecules of DNA in eukaryotic cells are linear, and then they are packed in discrete bodies we call as chromosomes. So here in this picture, we can see chromosomes that have been stained to fluoresce or glow, and then have been photographed so that we can see them And we can notice that they are actually discrete particles. They are separated from each other. They're not connected to each other. And these chromosomes have been particularly photographed at a certain stage of the mitotic or meiotic cell cycle, notably during meiosis, during the metaphase stage. So, scientists, after they take pictures of these chromosomes, they may then cut them and then arrange them in order according to their shapes, their sizes, and the location of their centromeres. That's the constriction that we see just in the middle of the length of the chromosome. And then we arrive with this photo we call a karyogram. Okay? So this is a karyogram. And we notice that all of those randomly arranged chromosomes a while ago are actually in pairs. And we call these pairs homologous chromosomes. Okay? So one pair of these, there are 23 pairs. One pair came from the male parent, and the other pair came from the female parent of this particular organism whose karyogram we have here. So this is actually a human being, okay? This is human karyogram. And we can see here in the human karyogram that although there are 23 pairs of homologous chromosomes, the last pair are actually quite special, and they are the sex chromosomes, seen here as X and Y. So this is from a male human being. And in the arrangement of the chromosomes that we saw a while ago, it was obvious that they were paired according to their shape and the location of their centromeres. Well, we can classify chromosomes according to just that one, the location of the centromere. There are chromosomes whose centromeres are exactly at the center of the chromosome, the length of the chromosome, and we call these chromosomes metacentric. And then during the anaphase stage of meiosis or mitosis, we can see here on the right how they would look like as the spindle fibers pull them towards opposite poles. Then we have chromosomes whose centromeres are located closer to one end of the chromosome. So as a result, we have a segment above the centromere that's shorter, and we call this the petite arm, shortened as a P arm, and a longer arm found usually at the bottom when the chromosome is arranged in an upright manner, and it's called the Q arm. And Q is really just there because it's the next letter after P for petite. So Q doesn't mean anything, just a next letter after P. Then we have chromosomes whose centromeres are closer still to one end. As a result, the petite arms now are a lot smaller as compared to the Q arms, and we call these chromosomes acrocentric. Notice how they differ on their looks as the spindle fibers pull them on opposite poles of the dividing cell here on the last column of this table. And lastly, we have chromosomes whose centromeres are located exactly at the end of the length of the chromosome. And they are known as telocentric chromosomes. So if you can go back to a karyogram as shown a while ago in the previous slide, you would notice that the homologous pairs are similar in terms of their location of their centromeres. So here is a more detailed look of a chromosome. In fact, a chromosome is actually made of supercoiled loops of DNA combined with certain types of proteins called histones. And they are looped so tightly together, the coil is so tight, that we can actually see already the structure under the microscope, of course, after staining. Thus, they're known as chromosomes. However, when these chromosomes are not yet fully coiled, they actually appear to be like cotton candy, to be as if they're randomly arranged inside the nucleus. And then we can call them there the chromatin fibers, as shown here on the illustration on the left. So the chromatin fibers will condense into a chromosome just before a mitotic or a meiotic type of cell division happens for easy distribution of the DNA, and then later on will become sort of disorganized again into chromatin fibers after the cell division. So the reason for the condensation of these chromosomes, as what we see here on the right, from what we see here in the left, is for organization purposes, for simple packaging of the DNA as they are distributed into 2 or maybe 4 new daughter cells later on. So we can see here the sister chromatids, which are actually the identical portions of the chromosome. They're identical based on DNA sequences. They are the same. They're attached to each other using a molecule called cohesin, which later on during the anaphase stage of mitosis or meiosis may be broken down by an enzyme known as separase. And then the centromere actually contains a portion called the kinetochore, illustrated here in purple. And that's where the spindle fibers used to hook on the chromosomes as they move during mitosis and meiosis attach. And then there's a special protein in the centromere region called the Shugoshin protein, which actually helps prevent the early degradation of the cohesin in that portion of the sister chromatids before the anaphase stage. So in the next videos, we will be looking more closer at the processes that happen during mitosis and meiosis.